Why do you think this type of research is of significance today? Why is it important to do that?
Well, my perspective as someone who works on microbes is that, in a certain extent, all of life is microbial. We are multicellular beings, but we are made up of individual cells, and those cells interact in very intimate ways with a whole host of organisms on and within us. So there's a very real sense in which we are walking, talking microbial growth chambers in a very strange way. My interests really go back to some of the earliest events in cellular evolution, and from the very beginning, I think it's true that cell biology is all about interactions, organisms interacting with other organisms. Symbiosis is a consequence of that. It is the natural order of things, and multicellular organisms, after they evolved, plants and animals, up through to complex beings like ourselves, really are a legacy of past interactions between different microorganisms. And there's a very real sense in which human biology is, our health and well-being are very much related to the way that our cells interact with the microbes within and around us.
Why is that now all of a sudden so interesting and such a hot topic in the biological sciences?
Well, there's been a number of revolutions, I think, that have converged and produced a situation where now is an amazing time to be doing this kind of science. With the realization, as I said a moment ago, just how intimately our well-being is connected to our microbial inhabitants is one aspect of that. We now have tools like genomics, metagenomics, the analysis of organisms that can't be seen and can't be cultured, but we can learn about them from their DNA sequences. Those are tools that are still fairly new, I would say, and are really taking off as a way to interrogate and ultimately understand the way our human biology is connected to the microbial biosphere.
Where do you see the field moving in the next decade? Where should it go? What is missing still?
There are things that we can certainly predict and some surprises that will undoubtedly come along. Certainly the field of metagenomics is in some ways still in its infancy. The technologies are evolving, and we're still learning how to collect genomic data and make proper sense of it. I think where we ultimately need to go, and where I think the field will eventually go, is a much more synthetic, robust, integrative approach to understanding the diversity of microbes that we interact with, how those microbes interact with one another, and how our own biology is impacted by and in turn influences the microbes themselves. I think it's going to be a two-way street there, and so I think that's sort of the holy grail in many ways, is to really understand the details of those interactions in both directions.
Why do we have to talk about these issues not only in our academic circles, but as you did it beautifully with your book, also with other means to reach the general public?
Well, I think it's important to tell the story of symbiosis and the importance of symbiosis to the general public, because it is the reality of our existence, and there is a myriad of ways in which the biology of symbiosis is truly relevant to human biology. Our own mitochondria, the energy-generating organelles within our cells, are remarkably derived from once free-living bacteria. So symbiosis is not something that is relevant in some circles but not others. It is quite literally a very part of the fabric that is a complex eukaryotic cell, and human beings are made of eukaryotic cells. So that's very fundamental. There's many aspects of human biology and medicine that are influenced by mitochondria, these energy-generating organelles. More and more, the discovery of diseases that are related to mitochondrial dysfunction. So there's a huge capacity to improve our understanding of human health and well-being by understanding our mitochondria in particular, but the phenomenon of symbiosis in general.
Thank you very much. Thank you.